away, right? Like that Star Wars title thing, right? It just disappears off into the thing. So I have written the exam, and I can tell you the format, I think, is absolutely identical in terms of numbers from the first exam. Well, very pretty close to it. So there's 20 points in the first section, there's 50 points in the second section, and there's 30 points in the last section. There are 24 questions in the second section. That's because 2 of the questions have 3 points each. So you'll see that. That's pretty straightforward. And I try to write it, and believe it or not, I really try to write them all the same way. You never can tell. I never can predict if one's going to be easy or hard or whatever. I'm the worst person in the world to ask, so I don't know. But I feel like it's representative of the material, and I would wager that it's about the same in terms of difficulty as the last exam. The one thing that's a little different from the last exam is there's more material here. I think you're probably feeling that as you're looking through stuff. A lot of heads nodding. I see that going on. Okay. So that's because we had that snow day on the first one, or the rain day, the flood day, I guess it was, that blocked out basically a day of lecture from before. Hopefully you've had some of these things like DNA replication and so forth in other classes that will help you as you're getting through here. All right, so the way I'm going to do this today is I'm just going to take questions, and as long as you have questions, I will be here. And if you run out of questions, then we'll call it a day. So whenever this will end, whenever you want it to end. We can't go longer than 50 minutes, though. So, yes, sir.
Can you talk a little about the equation with L equals T plus W in DNA replication?
Can I talk about the equation? I'm sorry? Sure.
What is the situation W?
Okay. So this is a common question. The question is, can I discuss the topoisomers and the equation L equals T plus W that I've talked about in class? So I'll be happy to go give you some additional information in there. And his question relates to, well, how can I tell about writhing and so forth? And so I've had a couple students come and ask me about this figure right here that they see. And the question was, and it's a very common question also, how do I tell if I have negative writhe or positive writhe? Okay? The answer is you're not going to look at this and tell. Okay? There is a convention whether it crosses over or it crosses under in terms of whether that's positive or negative. It doesn't matter for our purposes. You will always be able to tell positive or negative in this class based on the equation. The equation will always tell you, right? L equals T plus W. So if you have something that has 25 turns and you have something that has a linking number of 23, then that tells you that the writhe has to be negative. Has to be negative 2. Okay? Because that equation will always hold true. That equation will always be the case. All right? Now, what you see in the process of this is that when we have a closed circle, which is what we have in the situations that I've shown you, when we have the closed circle, the linking number is going to stay the same. Okay? The twists will vary. The twists will vary. Now, this is conceptually a little hard for me to describe to you in terms of retwisting, okay? And when I say something retwists, what happens? Well, literally, the rubber band twists itself. It's twisting, and because, remember, these bonds are single bonds, the phosphodiester bonds are single, and so they can rotate. And so this guy will literally twist itself a couple more times to relieve the tension that arises, and what causes the tension in the double helix. What did I say caused the tension? Supercoiling does not cause the tension. Supercoiling arises because of the tension. Well, you're getting close. It has something to do with the helix.
When you get away from the 10.5 base pairs.
Right. So if you get away from 10.5 base pairs per turn, that's what relaxed DNA has. All right? So if I change the number of twists so that the number of base pairs per turn isn't right, this guy's going to retwist if it can to get those twists where it was before. Well, there's a cost of that, and the cost of that is that the rest of the molecule has to in some way compensate for that, and that compensation happens in the form of writhing. Okay? We can't just make changes without there being consequences. The consequences of these changes, in the case of a closed circular DNA is writhing has to happen. Okay? Imagine taking a rubber band, okay, and twisting, one side of the rubber band. Does the rest of the rubber band respond to that? It has to. Okay? We can't put energy into something and have there be no consequence. There will be consequence of that. In this case, the consequence is that the circle will writhe. Does that help? Okay. So you're not going to have to recognize anything in terms of plus or minus or any of that. Okay. Understanding what writhe is, what twists are, and what linking number is important. Yes?
The type 2 topos.
Type 2 topos.
They cleave at the same spot. They cleave both strands in the same spot.
Oh, good question. So his question is, do type 2 topos cleave in the same spot? Do they have one strand here and one strand over here, or does it cleave across the two strands? It cleaves right across the two strands. Right across it, yeah. Type 2 topos are, I think, odd, as I said in class. They're odd and interesting in the sense that you've got to keep track of both strands. And the way that the cell actually accomplishes this is it passes through those strands another portion of the circular DNA. It's a mechanism that, again, words don't describe it very well. You actually have to see it in progress. But the number of things that the enzyme has to keep together in order to pass that enzyme through there without losing connection of the two strands is impressive. It's very remarkable what this enzyme does. And it does it very fast, because remember that the helicase is opening everything up at 6,000 RPM. The gyrase has got to basically be relieving tension at that same rate, right? If it doesn't, we're going to knot up our DNA. Okay, other questions? Yes?
What was the name of the enzyme that converts DTMP to DTDP?
Okay, so what was the name of the enzyme that converts DTMP to DTDP? Okay, so I'm going to give you a simple way of naming all of those. Okay, the general name of enzymes that convert nucleoside monophosphates to nucleoside diphosphates, whether they're deoxy or not. Okay, those are called. The general name is nucleoside monophosphate kinase. All right. Now, the specific one, if I asked you a specific one, then I would say this would be thymidylate monophosphate kinase. Or if you said thymine monophosphate kinase, that's fine. Okay. I'm not going to pick you apart in terms of that name. All right. If I went from GMP to GDP, it would be guanosine monophosphate kinase. Okay.
There's no difference between the
There are. Are there differences between the ribose and the deoxyribose? There are. Okay. But we're going to keep it simple. And the point is that the most important thing I want you to recognize, name of the base, it's a monophosphate kinase. Okay. So if you have those in there, you'll have the correct one. Yes?
Topoisomerases. It says that you have a portion of the DNA in the G segment, and you have a portion of the same DNA in the T segment, and it's passed through the break. In the, I don't.
Okay, you're talking about the gyrase now?
No, back to the topoisomerases.
Okay.
Type 2.
Yes. So that's what gyrase is. Gyrase is a topoisomerase type 2. Okay. So say it again.
And
And
it says that it's
it passes it through.
a segment and a T segment, and it's passing through the break.
Yeah. To be honest, I used highlights from earlier when in a different term where I've talked about that. I haven't talked about that this year, other than what I just mentioned right here, where you pass things through, and that's all it's referring to. Yeah. So don't sweat that. I do reuse my highlights, and it's get people concerned. Yeah, Giovanna. Sure. So let's go over the telomeres and telomerase and all that stuff. So that's a fascinating story. And so let's hit some highlights of these guys right here. All right. So first of all, let's get some nomenclature. So a telomere is a repeated nucleotide sequence at the end of eukaryotic chromosomes. Repeated DNA sequence at the end of eukaryotic chromosomes. The repeats are about 8 to 10 nucleotides in length. And it's repeated over and over and over. So that's what a telomere is. A telomerase The name A, the letters A, S, C at the end tell you it's an enzyme. A telomerase is an enzyme that makes the telomere. Okay? The enzyme telomerase, I said, is an unusual enzyme because A, it is what we call a reverse transcriptase. That means it is able to use RNA as a template to make DNA. Uses RNA as a template to make DNA. And that's important because telomerase carries its own template with it. Jody?
In that case, shouldn't the thymines on that template be uracils?
That is correct. That should be a uracil. That's not correct. Yeah, that should be a U right there. Yep. One of your books, several errors, and there's another one. Okay. All right, so these should be used because this is RNA. It's carrying RNA with it. Yeah. All right. Now, it's copying RNA, making DNA. The DNA it's making is on the 3' end of one of the strands. All right? So it puts this template up here, it copies, it lets the first two guys pair, and then the DNA polymerase says, okay, here's a T, I'm going to put, what actually is a U, I'm going to put in an A, there's a C, I'm going to put in a G, there's a C, I'm going to put in a G, another G, then a T, and then a T. Okay? So it's going to do that, and then it's going to slide down and do the whole thing over again and again and again and again. All right? What happens when that process has occurred is, if we think of this top strand now, it now, instead of ending right here, it ends way over here. All right? So now we've got this long 3' end. We've got to get to 5. How do we get that? Well, remember that a 3' end can be copied if we have primase. Primase can start it. So let's say the primase comes at the very end of this and it lays down a little RNA primer right there, moving back now in the other direction. DNA polymerase says, oh, there's a primer. I can elongate the primer, and it fills that whole business in. But what's happened in this process is we have elongated the end. Only the very far 5' end is going to have that little RNA primer. Okay? So instead of having a telomere that ends over here, we have a telomere that ends way over here on your right. Okay? So the telomerase has built a longer linear end. Yes, sir?
At the very terminus, there was some discussion that it has the single strand that loops around on itself, right?
You're talking about it at the far terminus up here?
Of the telomere, yeah.
Yes, yes. That confuses the picture, but yes, that's right. So, go ahead.
Which model are we going with? That the primase goes as far out as possible?
Okay.
Or that it loops?
Okay. So what Jody's talking about is the looping process actually helps to explain how the other strand gets made. And while that's true in some cases, like HIV virus and things like that, that's not something that I want to go into here. So what I'm telling you is that you get elongation here, and then primase, which brings the rest of it back. Okay? Okay. Now, telomerase, as I noted, is present in fetal cells. It is important for lengthening those telomeres. And some people think, and there's some interesting evidence that suggests that the lifespan of those cells and or the organism may be determined, at least partly, by the length of the telomeres that are there. Now, there's some really cool stuff that people have studied with this. And what they include are comparisons of the longevity of, for example, children born from a father who was 80 years old. If you think about it, that kind of is an interesting story, right? They're starting out with shorter telomeres, okay? So even if everyone gets lengthened to approximately the same length, does that mean that if your father was 80 years old, that you would have a shortened life expectancy? What do you think?
I might assume the inverse.
You might assume the inverse.
If he lived to reach 80, he probably started out with some pretty high set points.
No, I'm talking about the children, not him, the children. But you're still, oh, I see. You're saying because he had other factors besides the telomeres that gave him longer life, okay? You said no. Okay? Nobody says yes. To me, that's a logical thing I would wonder about, right? They're starting out with a shorter telomeric length, and if that is a factor in longevity, then we might expect that the children of very old men would have a reduced life expectancy. At least I would, all right? And, Connie?
But doesn't he only get half, isn't it the child who only gets half of his or her genes from the father? So
Child only gets half of the genes from...
telomere would be like relatively at a normal length, like from the mom. I mean, doesn't that...
Right, but still, some of the genes of the father are going to be lost when that telomere disappears, right? And if those are necessary genes, let's say mom has a trait that is no good, you're still going to need those father's genes. So, no, that's a concern. Anesia?
During spermatogenesis, don't those come from stem cells?
So, yes, during spermatogenesis, there is a lengthening of that telomere, but I'm saying if that lengthening is an average, average length, let's say every time that happens, we put on 1,000 of these, okay, then if it starts shorter, you would expect that the product of that would be a shorter telomere that would be in those children. And I'm not the only person who's asked this question. There's other people who have asked the question, and they've done studies, and the upshot is that the studies suggest that there's not a major contributing factor. Not a major contributing factor. So maybe what Jody said, this person has good genes for other things, perhaps that is a bigger factor in longevity than telomeric length. Okay, so, yep.
I was assuming more along the line that the telomeres hadn't been completely chewed away and we're losing actual genes now, but that the father, since he lived to 80,
Oh, I see,
had
okay.
long telomeres to
The
start,
father had longer telomeres to start.
and
Yeah.
that genetic gene could be passed on so
Yeah.
that the progeny had longer ones to start.
You could imagine that this would be there'd be a lot of variables in something like this, and that's part of what you're pointing out is true. One of the reasons people asked this question originally, and it's a real concern, is if we think about cloning animals. Okay, Dolly the cloned sheep, all right, she was made from an adult cell, okay, an adult cell. So this adult cell had gone through many rounds of replication, had shortened telomeres to start. Did Dolly die at an early age? She did. And she died of exhibiting symptoms of old age at an early age. And so this is what really got people thinking that, oh boy, this may be a real factor if we're trying to clone organisms. And the picture is not completely clear. Some organisms appear to exhibit exactly the same traits as Dolly. They die early, they get old early, etc. And others, there's no effect whatsoever. So the answer isn't completely clear. And there's probably more to it than simple telomeric length. But it's a reasonable question to ask. Okay, back to answering questions. I'll shut up. Connie?
What prompts RNR's tyrosine to become a radical?
What prompts RNR's tyrosine to become a radical? Okay. Ultimately, what's happening with RNR's tyrosine to become a radical is that there's electronic shifts that are made as a result of that NADPH donation that I talked about before. Okay. And so ultimately things are happening arising from that. But it's a very long story to get to that point. Very long story to get to that point. Karen?
In fatty acid biosynthesis, you use the names malonyl ACP
Yes.
and malonyl CoA.
Yeah. Is there a difference between malonyl ACP and malonyl CoA? Absolutely. All right, so let's think about this. Let me show you the stuff. So we have to, and both of these exist. They both exist, and we have to consider both of them. So I'm glad you're asking the question here. If we look at fatty acid synthesis, you may recall that the enzyme acetyl-CoA carboxylase catalyzes what I described as the first step in the synthesis of a fatty acid. You'll notice that there's a CoA on the end of that guy, okay? And the name of the enzyme tells me that there's a CoA on there as well, acetyl-CoA carboxylase, right? That means that my starting template is an acetyl-CoA, all right? The product of that is a malonyl CoA. But when I go to start putting pieces together, the pieces get put together with an ACP. That means that all of the CoAs have got to be replaced by ACPs. All right? So when I go to make my very first joining of two fatty acids, let's go to here. You'll notice that by this point, I've got an acetyl ACP, meaning I've taken an acetyl-CoA and I've put an ACP on it. I've also taken a malonyl ACP, or malonyl-CoA, and put an ACP on it. So yes, they're both real and they're both there, and they're necessary. So the CoA carboxylase requires CoA. Make sense? Yes?
In the conversion of triglycerol to fatty acids, how much of the details of that reaction do we need to know?
So when I talked about the breakdown of tricyl triglycerides, basically, okay, so the triacylglycerols, which are fats, breaking down to fatty acids, what have I talked about with respect to that, okay? So let me just show you briefly what's there. Okay. So if I go from that's not what I want. Fat breakdown. Okay. All right. So this schematically shows the breakdown of a fat. Now, I may not have been real clear, so if I wasn't, I'll try to be clear here in terms of what this process is occurring as. Okay? This process can occur inside of cells. This process can occur outside of cells. So if I take fat in my digestive system and I eat some fat, I talked about how it had to move across the intestinal wall. And I said the first thing that happened was there's an enzyme that takes off a couple of fatty acids, and then it gets moved across and the fatty acids get put back on. Okay? Those processes are happening outside of cells. They're not occurring inside of cells. They're happening outside of cells with secreted enzymes called lipases. In fact, all the enzymes that break down fat are called lipases. So as a category, that's what they're called. All right. So you're seeing here a lipase taking one of these guys off. You're seeing a lipase taking a second one off. And you're left, in this case, with a monoacylglycerol, meaning a glycerol that's got one fatty acid on it. This would get moved across the membrane, moved across the intestinal wall into the lymph system where the chylomicrons would package it up. It would get put back together as a fat, and it would leave at that point. All right. The other process I talked about occurs inside of cells, and it's the process inside of cells where there's some regulation. And it uses lipases as well. Okay? The lipases that happen inside of cells, okay? So we're seeing now we're inside of a cell. What we're seeing is that the very first enzyme that takes off the very first of the fatty acids, all right, is called triacylglycerol lipase. It's this ATGL that's here. All right? Triacylglycerol lipase is the only enzyme of fat breakdown or fatty acid breakdown that is regulated. And it's regulated hormonally. Now, I haven't gone through all the perilipin and all that business there, so I haven't talked about that. But I have said that the triacylglycerol lipase, which cleaves off that very first fatty acid, is, in fact, regulated. Okay? Once that has been broken off, then other lipases will take off the other two fatty acids. Remember, we've got 3 fatty acids on a fat. Only the first one's coming off is regulated. That's regulated by triacylglycerol lipase. Right? And then everything else comes off. All the breakdown, all the beta-oxidation, no regulation. It just occurs. It just goes and breaks it down and goes off and makes energy. Does that answer your question? Liz?
Several questions. One is, do we need to remember this? Is it the beta-adrenergic pathway to get to
Yeah.
phosphorylating?
So the question is, am I going to make you regurgitate the beta-adrenergic pathway? I don't think that would be productive at this point. I think you should know that it is a beta-adrenergic receptor that's involved in that. Yes?
And does the triacylglycerol, does that take off the first fatty acid extracellularly as well as intracellularly?
Okay, so it's a very good question here. The triacylglycerol lipase that's hormonally regulated, is that both intracellular and extracellular? No. No. Okay. So the triacylglycerol lipase that I talked about before, the extracellular one, is just floating around in the digestive fluids and so forth, and it's not regulated because it can't be. This process here has to occur inside of a cell. So it's a different triacylglycerol lipase, and it's not the same one as we have here. So only this one inside of the cell will be hormonally regulated.
Last
Okay?
So how do the bile salts interact with
Yeah.
this?
How do bile salts interact with this? Well, okay, so it's a very good question. Bile salts are necessary to emulsify fat to facilitate two things. One is the transport across the membrane. Okay? The other is to just sort of make a water-soluble thing that these enzymes can act on. Okay? So the enzymes are going to be water-soluble, and the enzymes have to have an ability to interact with something that's not water-soluble. So a detergent in the form of bile salts facilitates that interaction and allows the triacylglycerol lipase to have access to the fat. Make sense? Yes, back there.
In the fatty acid oxidation, I just use it 7 cycles of beta-oxidation of HMG-CoA to make your glucose.
Yes. Okay, so the question was, if I have Pyrimidine-CoA and I break it down, how many cycles does it take? Well, it turns out it takes 7 cycles, because each cycle breaks off 2. And you say, well, there's 16 carbons. Shouldn't it take 8 cycles? Well, no. Remember, the last one splits 2 in half. Right? So the last one, when you cut the last one, you get 2 out of that one. Each of the other breaks gives you 1. So I have 6 breaks that give me 6, and 1 break that gives me 2. I get a total of 8 pieces. Does that make sense? Yes?
In urine synthesis, is the SAM protein, the SAM, is it considered a folate because it's donated to the metabolic?
Okay. So the question is S-adenosylmethionine, which is a methyl donor, considered a folate because that's what folates do? The answer is no, it's not. It's completely separate from a folate. Connie?
Ndpk, you said it can change all triphosphates into triphosphates using other triphosphates. Can it like mix and match between ribose and deoxyribose?
Yeah, so good question here. Connie's asking about Ndpk, and I said that it can convert all of the nucleoside diphosphates into nucleoside triphosphates, and it can use nucleoside triphosphates as energy sources to do that. Her question is, can you mix and match virtually anything and everything there? And the answer is yes, you can. Yeah, it's very flexible. Now, one thing I haven't told you, but I'll tell you because this may make you a little confused. Remember I said the importance of balance. I want to balance how many of the ATPs I have, the GTPs, and so forth. So is this going to upset all of that balance? Okay. The answer is that Ndpk has some preference for one or the other of the triphosphates. So that's built into the enzyme and actually helps establish the balance as well. So technically, Ndpk is a balancing enzyme. I have not talked about that in this class, and I'm not holding you responsible for that. But yes. But the answer to your question is yes, mixing and matching of anything and everything is possible through Ndpk. Yes?
So are foam cells kind of precursors to atherosclerotic plaques?
Yes. So our question is, are foam cells precursors to atherosclerotic plaques? And the answer is absolutely, they are. Foam cells arise when we have the immune system attacking what it perceives as damage to an LDL, to something that the LDL is linked to. Perhaps the LDL got stuck to the artery because of a chemical reaction that occurred. The immune system attacks that, and it's the building up of more and more of the immune components there that gives rise to that foam cell. Yeah. Liz?
In your notes, in class, you mentioned that smokers have more free radicals.
Yes.
Also, in your notes, you mentioned that smoking could increase LDL.
Yes?
Is that both?
So the question is, I said that smoking can increase free radicals. I also mentioned that smoking can increase LDLs. Is it true that smoking can increase both of those? The answer is absolutely yes. Yeah. And they get a double whammy. Smoking gets you, we've already seen about 4 or 5 whammies this year. So smoking gets you in many ways. Yes, question back there.
In pyrimidine biosynthesis,
Yes?
does it always go to UMP and then UMP goes to CTP and TP?
Okay. So her question is, in pyrimidine biosynthesis, first of all, is UMP the first thing that's always made? The answer is yes, in de novo synthesis. Second question was, does it always go to CTP? And the answer is yes, so long as there's not already too much CTP. Okay? So UMP goes to UDP, goes to UTP, and then goes to CTP by cytidine synthase, right? Okay? So the answer is yes, that is true. And the other question was, I think, does it also go to DTTP? And yes, it will, but remember the pathway that it gets there. It gets there by starting with UDP, not UTP. UDP goes to DUDP, that's ribonucleotide reductase. Goes to DUTP, goes to DUMP, goes to DTMP, DTDP, DTTP. Okay? It's a convoluted pathway to get to T, but it goes through the uridine nucleotide. Okay? Yes, Connie.
How much do we know about the details of the IMP synthesis? Because there was a really nice, big, complicated mechanism you showed on the screen. You said you don't really know it that well, but you also explained some parts of it.
Well, so the question is how, what is relevant of the IMP synthesis that I talked about? The answer is, as always, whatever I talk about is fair game. All right? I'm not going to go ask you structures. I'm not going to go ask you anything detailed in there. But if I talk about an enzyme on the way, then that enzyme would be a relevant enzyme. And so it's the same rule as always. That is, if I've talked about it, it's fair game. Okay? Liz? Oh, okay. So what's the difference between isoprenes and isoprenoids? It is a semantic difference, and the difference is this. An isoprene is a specific general structure. Does that make any sense? A specific general structure. That is, it's a 5-carbon building block. When I use the term isoprene, I'm talking about a 5-carbon building block. I mentioned 2 5-carbon building blocks in class, dimethyl allyl pyrophosphate and isopentenyl pyrophosphate. So each of those is an isoprene. All right? An isoprenoid is something that I make with an isoprene. So anything bigger than 5 that I make with a 5-carbon component would be an isoprenoid. Geranyl pyrophosphate would be an isoprenoid. Cholesterol would be an isoprenoid. Squalene would be an isoprenoid. So anything I've used isoprene units to make becomes an isoprenoid. Does that make sense? So an isoprene will always have 5 carbons. An isoprenoid will always have more than 5 carbons. Yes, Karen.
When you're discussing salvage pathways, you say that nucleic acids can be broken down to nucleoside monophosphates, and then you say monophosphates can be rephosphorylated to triphosphates. Are those still nucleoside monophosphates?
That's correct. So in I think the notes I said that nucleoside triphosphates can be broken down to nucleoside monophosphates, and then I said monophosphates can be built back into triphosphates. Was I referring to nucleoside monophosphates? And yes, I was. I was just being brief. Yeah. Yes, back there.
So when you're referring to nucleoside monophosphates, that's because nucleoside.
I'm sorry, I heard the first part. I didn't hear the last part of what you said.
So when you refer to nucleoside monophosphates, they're nucleotides.
Nucleoside monophosphates are the same as nucleotides. That's correct.
So would you prefer for us to refer to them as nucleoside monodiphosphates or nucleotides?
Well, it depends on what I'm asking you. So if I asked you to name or I said to what is ADP, I would in general hope that you would always most accurately answer what I'm asking you. It would be a nucleoside diphosphate, right? If you say nucleotide, that's correct, but it's not as specific as nucleoside diphosphate. So it always depends on what I'm asking. Yeah. Yes?
I'm not clear about the reactions at the mitochondria where you've got acyl-carnitine and carnitine and acyl-CoA, why that is happening.
Okay. So it's a question about where does carnitine fit in this bigger picture that I've shown. So let me show you that. What's happening? Why do we have carnitine and what's it doing? Okay. So metabolism of fatty acids happens inside the mitochondrion. And fatty acids come into cells by a variety of mechanisms. But when they arrive inside of the cytoplasm, the first thing that happens is they get a CoA that's put onto them. that makes an acyl-CoA. So for example, palmitoyl-CoA is an acyl-CoA. It started out as palmitate. A CoA got put onto it. All right? As it's traveling to the mitochondrion, when it gets to the mitochondrion, the CoA on the end is not a structure that the mitochondrion can transport across the membrane. So the CoA has to be taken off and replaced by something else. And the thing it is replaced by is this structure called carnitine. All right? And so you see that happening here. Here's the acyl-CoA that is coming in from the cytoplasm. The acyl-CoA is having the CoA removed, and it's being replaced by a carnitine. So where we had acyl-CoA here, we now have acyl-carnitine. The reason for doing that is the mitochondrion does have an antiport that will bring an acyl-carnitine in. It won't bring an acyl-CoA in. So the acyl-carnitine makes it into the mitochondrial matrix, and then the reverse of that process that you just saw happens inside the mitochondrion. So now the carnitine is replaced by a CoA. We get an acyl-CoA out, which is what we started with up here. And carnitine is kicked off of it, which now is kicked back out to complete the cycle. Okay? This acyl-CoA then becomes the substrate for beta-oxidation. Does that make sense?
Is that because of size problems?
So is that because of size problems? It's actually it has to do with the specificity of the protein that's doing the transport. So every protein that does transport has very specific dimensions of things that it's going to move through. A glucose transporter is going to move glucose. It's not going to move alanine across a membrane. Okay? So this guy has evolved a preference for carnitine on there, and that's why you see that swap. Okay? Yes, Connie?
I have a quick question about that figure.
Okay.
On one side it says the cytoplasmic side, on the other side it says the matrix side. What happens to the inner membrane phase?
So the question is, all right, so here's the cytoplasmic side, here's the matrix side, and where's the intermembrane space? Okay. Well, the cytoplasmic side doesn't mean it's the cytoplasm. It means that's the direction of the cytoplasm, right? So this actually is in the intermembrane space. Yeah. Anesia? The things that affect the activity of ribonucleotide reductase, you're thinking about the activity site and the specificity site. Okay, let's talk about that. So if we go back to nucleotide metabolism, and I bring up my DNTP synthesis, okay, here is the regulation, which I think is actually down here. Okay, this shows the large subunit, and I said it was the small in class, and so I did correct that, and so it is the large subunit where all this is occurring. The large subunit has on it two different binding sites for nucleotides. Two different binding sites for nucleotides. All right? One binding site is called the activity site. That's up here. The other site is called the specificity site. And technically there's three because at the active site, the place where the reaction is catalyzed, that's where the substrate binds. And the substrate, of course, is a nucleotide as well. Well, when we think about the activity site binds one of two different things. It binds either ATP or DATP. These have exactly opposite effects on the enzyme. ATP turns the enzyme on. DATP turns the enzyme off. If cells have too many deoxyribonucleotides to start with, there's no reason to make more. So a high level of DATP turns the enzyme off and stops additional synthesis of deoxyribonucleotides. Okay? If cells have an abundance of energy, they've got a lot of ATP sitting there, you want to use that energy to do something, you're going to replicate and divide, you want to make deoxyribonucleotides so that you can make DNA. All right. So those two are straightforward. The allosteric site, also called the specificity site down here, and yes, this is an allosteric site as well, but this is called the specificity site. The specificity site I simplified, okay? And I simplified simply to point out to you that this binding site right here determines what the enzyme wants to work on. So it's not turning the enzyme on or off. It's changing the enzyme's preferences for what it's going to bind to. The binding is going to occur here. So the example I gave was if I have plenty of DGTP, DGTP is a purine. It means I've got too many purines, I need some pyrimidines. What's going to be favored binding here? Pyrimidines. That's going to happen in the form of CDP or UDP, which are both pyrimidines. If I have an excess of DCTP and it binds here, then what's going to happen is this a pyrimidine, this is going to favor the binding of purines, and that's going to favor the binding of GDP and ADP. Now, that's a simplification. It's actually somewhat more complicated than that, so I don't mean to take that as verbatim truth if you go work in a nucleotide metabolism lab. But in general, that's what's happening with this enzyme. Okay? So this guy is simply controlling what's binding at the active site. It's changing the enzyme's specificity, its preference for substrate. And as a consequence, it's having a great influence on how many of each of the deoxyribonucleotides are being made. Does that answer your question? Connie?
Sorry, quick clarifying question. The activity site, will it bind to only ATP and DATP, or can it bind to other nucleotides?
The activity site only binds to ATP or DATP. Yes?
In the control of HMG-CoA reductase, you listed 4 different types of controls: the solitary degradation and covalent modification. Can you speak to which things are specifically controlling each of those?
Okay. So her question was, when I talked about the regulation of HMG-CoA reductase, I talked about 4 different ways of controlling how active that enzyme is. And her question is, can I say how each of those is controlled? Okay? Well, yes. So let's think about what those 4 mechanisms are. First of all, we have feedback inhibition. What's the feedback inhibitor of HMG-CoA reductase? Cholesterol. Okay? Second, we have control of whether or not the enzyme is made. Anybody remember how that was controlled? So there's a transcription factor called SREBP, and I gave it the designation C, SREBP-C. That was the controller of whether or not transcription was going to occur. Okay? Third mechanism was the rate of degradation of the protein. Okay? What bound to the protein and put ubiquitin on it? INSIG. Very good. And the last regulation had to do with its covalent modification. Anybody remember what the modification was? Phosphorylation. So how do we put phosphate on? Kinase. How do we take phosphate off? Phosphatase. Okay? That makes sense? Yes? Do kinases use ATP to put phosphates on? Yes. Kinases almost always use ATP.
I remember you said that HMG-CoA reductase will basically cholesterol synthesis stops when ATP levels are low.
Mm-hmm.
So how does that work?
Okay.
Does it just still get ATP?
Okay. So question was that I said that cholesterol synthesis doesn't occur much when ATP levels are low. How is that regulated? There's several ways in which that can occur, but the easiest one to understand is since ATP is a substrate necessary to make, let's say, geranyl pyrophosphate or farnesyl pyrophosphate, if that substrate is low, the reaction that's catalyzed involving that substrate is going to be down as well. So it just slows down because of that. Okay. And did you have a hand question?
I just wanted to clarify the HMG-CoA reductase, the phosphate turns it on or off?
That's a good question. Let me think about this. Let me think about it in sheet. I don't recall off the top of my head. I'll have to look. HMG-CoA reductase would be in here, I believe. And that would be it's in here, I think. No, it should be ketone bodies, fatty acid synthesis. Yeah, let's see. Let's go back. It's up here then. Here we go. Okay, HMG-CoA. I don't recall. I can't, and I don't want to tell you the wrong thing. So I would say look in my notes. I can't recall off the top of my head. Yeah, it's in there. I know it's in there. So. Yes.
How much detail do you want us to know about the glycerophospholipid biosynthesis pathways?
Glycerophospholipid biosynthesis pathways. Well, as always, whatever I talk about in class is fair game. I talked in general terms about two ways of making glycerophospholipids. I talked about activated intermediates. All right? I said we could have an activated intermediate that activated the diacylglycerol part, where I attach a CDP to the diacylglycerol, make a CDP diacylglycerol, that's activated. And then I could attach a smaller molecule to it, like inositol, and make phosphatidylinositol, right? That's one way of doing them. The other way I talked about doing them was I can activate the small molecule. What if I make phosphatidylethynolamine? I'm not phosphatide. I make CDP ethanolamine, right? So I've activated the small molecule. I can attach that to a diacylglycerol and make phosphatidylethynolamine, right? So those are general things. I'm not asking you for specifics in terms of, draw the way in which I make choline step by step. I'm not going to do that, okay? But the general process by which you make those, you should know. So, yes? I can't hear you. I'm sorry. Can I talk about when SAM is used? Actually, SAM is actually used in the process of making phosphatidylcholine. You start with phosphatidylethynolamine, and I will show you that very briefly. That was steroid and lipid metabolism, and that was here. Okay, so here's phosphatidylethynolamine that I just described one way of making it. I add to that 3 methyl groups from S-adenosyl methionine, and the donation of those 3 methyl groups are seen right there. That makes phosphatidylcholine. Okay? I hear rustling. So before you go, a couple things. I'm not going to be here. Dr. Merrill will be here. He will expect you to sit as I asked you to sit before. So please pay attention. It'll simplify things immensely. I want you sitting every other seat. Number 1 here, number 3 here, etc. Please pay attention because he's not going to be as nice about this as I might be. Okay? 1 here, 3. If somebody comes and sits in number 2, don't sit 2 over in number 4. Sit in number 3, and the person sitting in the even number one is going to have to move. This is true upstairs as well. Down here, number 1, number 3, 5, etc. So sit in the odd numbered ones as I've indicated. I'll be back on Monday and see you guys then. Thank you.